Imagine this, it's 3 a.m., your biochemistry exam is tomorrow, and you're stuck on the most important question, calculating the stoichiometry of a cellular reaction. Sound familiar? Don't worry, I've been there too. Today we're discussing stoichiometry in cellular reactions. Stoichiometry isn't just chemistry's evil cousin, it's actually the secret sauce behind understanding how cells work. By the end of this video, not only will you get it, but you'll also wonder why it seemed so confusing in the first place. Let's dive in. So, quick refresher, what even is stoichiometry? In simple terms, it's about balancing the inputs and outputs of a reaction. But in cellular reactions, it's not just numbers, it's about molecules your life depends on. ATP synthesis? Glycolysis? Even your favorite metabolic pathways, they all use stoichiometry. Think of it like ordering chai at a tea stall. You bring sugar and milk, the chaiwala uses tea leaves and water. Stoichiometry is the recipe that ensures no one gets a watery disaster instead of chai. Now that we know what it is, let's see why stoichiometry actually matters. Okay, here's the fun part, why should you care? In biotechnology, stoichiometry helps us design bioreactors, optimize metabolic pathways, and even understand how much glucose a cell needs to crank out insulin. I still remember working on a lab project where we had to calculate the exact ratio of oxygen to glucose for yeast fermentation. One wrong calculation, and instead of ethanol, we got a smelly failure. Not fun. But here's the thing, mastering stoichiometry isn't about memorizing, it's about thinking like a cell. And to do that, we need to tackle mole balances. Now, let's break it down, mole balances. If you've ever wondered, how much oxygen does 1 mole of glucose need in glycolysis, this is where the magic happens. In this reaction, stoichiometry tells us that 1 glucose molecule demands 6 oxygen molecules. The ratios are non-negotiable, it's like making biryani without rice. No. But wait, real cells aren't perfect, so how do we deal with real-world complications like inefficiencies or competing pathways? That's next. Here's the tricky part, cells don't follow textbooks. For example, in fermentation, glucose might produce ethanol and CO2, but not always in the same ratios. Why? Enzymes are choosy, environmental conditions vary, and sometimes the cell's just moody. So, how do we simplify these messy realities? The answer lies in tools like stoichiometric matrices. This is where it gets exciting. To handle complex reactions, we use tools like stoichiometric matrices or software like MATLAB. These not only balance equations, but also predict yields and help design experiments. For instance, in metabolic engineering, matrices let us optimize how much lysine a bacteria can produce, critical for industries like pharma and food. But wait, what if you're stuck without fancy software? Can you still ace this in your exams? Of course, you just need the right strategies. Alright, let's bring it home. For your exams, follow these steps, balance the simplest molecules first, e.g. O2, H2O. Double-check atom counts on both sides. Keep a calculator handy for mole conversions, it saves time. Finally, don't just learn stoichiometry, practice it. Whether it's solving equations or designing bioreactor models, the more you do, the easier it gets. If this video helped clear things up, hit that like button to let me know. Subscribe for more biotech hacks, because we're tackling topics your textbooks won't explain like this. Now, go ace those equations. See you in the next video, happy studying!